Now, at the beginning of our work in a manner of main topics of this training, I would like to invite Leif Skot to present us a lecture about genetic markers in plant breeding. Leif is principal investigator in Institute of Biological, Environmental and Rural Sciences and Aberystwyth University, United Kingdom. He has finished PhD in Plant Physiology and Agriculture Research Department in RISO, National Laboratory and University of Aarhus in Denmark. Now he is a leader of forage plant breeding team. His research is focused on association genetics and genomics assisted methods that underpin IBER's forage crop breeding program. Also, he works on transnational genomics with a view to facilitate the increasing in the rate of genetic gain for key traits in forage crops. Genetic mapping, steep discovery and detection, analysis and exploitation of genetic diversity in forage crop species in the breeding programs. He is also a member of Biotechnology and Biological Sciences Research Council in UK and a member of editorial board of the journal Scientific Reports. He is also leading one work package 1 in EUCLIG. So, Leif, please.
Thanks. Thank you very much for that, and it's a pleasure to be here. And it seems also very appropriate that we are in this city talking about using genetic markers in plant breeding, because this was where Gregor Mendel did his famous experiments and discovered the basic rules of genetics, the ones that we still use today. And it strikes me that what we are all trying to do is to sort of Mendelianize everything in terms of looking at associations between phenotype and genotype. We try to break everything down into small things that are inherited in a Mendelian way. I will go on to talk a little bit about genetic markers in plant breeding. It's going to be a little bit of a general talk, and I'm using some of the things that I've also used in a lecture I give at the university in Aberystwyth. You'll have to forgive me, some of it is too basic for you, but the structure of the talk will consist of these 4 main items. I'll talk about QTLs and some examples of use of molecular markers in plant breeding, and then a few words on genome-wide association studies and genomic selection. You will hear much more about that later on in the course. But since markers are used in those 2 fields mainly now, I could not mention it. Anyway, there has been a number of different markers over the years, but the ones I'm going to talk about today are only the single nucleotide polymorphisms that are sort of taking over everything almost, and SSRs, which are simple sequence repeats, so sort of from 2 to 6 bases of repeats, and they're very polymorphic and multi-allelic. So they are useful for certain things still, and there are some examples I'm going to mention which use simple sequence repeat markers, but mostly things are done with single nucleotide polymorphisms. Now, what has QTL's quantitative trait loci ever done for us, to rephrase a famous quote? So they have provided a basis for a lot of scientific literature, and they have also provided arguments for a lot of grant applications. Where you say you have this QTL and you want to look at the genetic basis of a certain trait, and over the years that has been used a lot, and it has actually provided relatively few examples of incorporation into crop breeding. But it is changing a little bit now, especially with GWAS and genomic selection. I think this is where even the breeders think that this is actually something useful. So this is a little overview of, I just took all publications, not just plants, but everything from Google Scholar that had the word QTL in it. And it started with 612 publications in 1980, and then in 5-year periods it escalated to over 50,000. It has dropped back a little bit, but that may be because we only got 4 years of data there in the last 5-year period there. So yeah, there's a lot of literature on QTLs out there. Now, if I can go back to the breeder's equation and look a little bit on that. I'll go through something that is from another course that's held in the UK called quantitative methods in plant breeding. That's led by a guy called Ian Mackay. And like me, he's a little bit of a worrier. So he went back to the breeder's equation. So the change in genetic gain is related to selection intensity, the genetic variation in the material you're working with, and the length of the breeding cycle, as well as and then heritability. And the use of marker-assisted selection has to increase the response to selection per year or per unit cost in order to be useful for the breeder. And this is taken from his notes. So if you select on phenotype, this is the equation. This is strange. And you can sort of rewrite it a little bit so it goes, so there's phenotypic heritability and the phenotypic variance. And if you select on markers, he then split the heritability item to heritability related to markers and the genetic correlation between a score for the marker and the phenotype. In order to give a greater response than phenotypic selection, this item has to be larger than that, and then you can reduce that by doing a little bit of math, and you basically come down to the squared genetic correlation between the marker index and the phenotype must be higher than the heritability of the phenotype. So that's quite a harsh, oh, sorry, I better go. That's a little bit of a harsh lesson there. So if you want, I mean, if you have QTLs that are perfectly aligned with the marker that explains all the QTLs, a marker at each of the QTLs that underlie the trait, and explains it totally, then marker-based selection will be unbeatable because the accuracy will be 1. But if it only explains like 50% or something, then it's up against a heritability, for example, of 0.5, which is not that unusual in many traits. So it'll have its work cut out to be useful in marker in breeding. So that's from Ian Mackay's course. Oh. Okay. Now I will go towards marker-assisted backcrossing, which is an example which is of a breeding or a crossing program where marker-assisted selection actually has been useful and there are many examples of that, particularly in rice breeding, I think. And so you want to integrate one or more loci from a donor parent to a recurrent parent. And you want to otherwise recover as much as possible from the recurrent parent of the genome, because from the donor parent you can risk there's a lot of sort of rubbish that drags down the agronomic characteristics of your crop. So you want to do foreground selection, so you need a marker that's closely linked to the trait, and where with markers probable heterozygotes can be selected, and that are particularly useful if the trait is recessive. Now, for background selection, you need to recover the recurrent parent genome by selecting for homozygosity marker alleles from the recurrent parent. So, here's a sort of a traditional scheme that I've taken from this paper here. You can find by going to that site there. And so you cross your two parents, and you get an F1, and you do foreground selection, for example, with an SSR marker that is almost on top of the trait QTL, and then you do back crossing to the recurrent parent. And in the first, in the F1, you have 50% of each genome, and then as you move along, you doing foreground and background selection with SSRs, you recover more and more of the recurrent parent as you go through the generations, and eventually you come to a stage where you have to do selfing to get the trait in question in a homozygous state. So you fix it, and then you go on from there and get your improved variety. So that's a basic strategy of so for foreground selection, you have the probability of the introgress trait is still there. That's 1 minus the recombination frequency. So if you, for example, go 4 generations down the line and you have a recombination frequency of 0.05, which is low, but still there between the marker associated with your QTL and your then after 4 generations, the probability of a recombination is 0.81. So there is it has dropped somewhat. So you can risk, if the marker is not right on top of your trait, is actually the causative polymorphism, then there is a slight risk that you're going to lose it if you just go by marker-assisted selection. So if you have a sample size of n, the probability of no individual carrying the haplotype is 1 minus half of this 1 minus the recombination frequency to the, and the n is the size of the population. So you can work out how big your sample size needs to be in order to have a certain, to be below a certain risk or probability of having having the gene not being there after a certain number of generations. So in background selection, so it's the probability you gradually, if you don't do any selection and you keep back crossing, there is the traditional equation that's a half to the nth, which so you go from 50% to 75% to 87 point something and so on. So after 6 or so generations, you are well up in the 90s of recovery of your recurrent parent. So according to Ian Mackay, he thinks that 2 to 4 markers per chromosome is considered sufficient to ensure recovery, and that could save a couple of generations of back-crossing using marker-assisted selection. So that's examples of where it is useful to use marker-assisted selection. And so background selection on the chromosome where your marker of interest is, it's a little bit more complicated, and I've just taken this from Ian Mackay's. So you want to reduce the linkage drag on that chromosome, so recover as much as possible over the recurrent genome, but still have your marker there you're associated with the trait. So, so if you have two markers flanking the QTL of interest, and the donor genotype is this, and then the recurrent parent is this, so you want to select for this, and then the probability of not finding this genotype in a population of size n is that, so you can calculate, again you can calculate that based on recombination frequency and the risk you want to run, how big your population size needs to be. If you go back to then recurrent selection, which is what I'm mostly used, well, most breeding, I guess, is some form of recurrent selection, but particularly in forage crop breeding, you are using recurrent selection where you do some crossing and you look at the progeny, and then you go back and use the best of the progeny in the new cycle. So that's the basic principle of recurrent selection, and that's what's used a lot in outbreeding crops such as the red clover and the alfalfa that we use. That we use in the UCLAC program. So if you go back and revisit the form of the breeder's equation, so the response to selection is related to heritability and the selection pressure that you put on, and so that is basically the difference between the mean of the proportion you have selected and the mean of the population you started off with. And then the response is the mean in the next generation, the C1, minus the mean of the original population. And then there is a sort of a conversion to a standardized selection differential called KP, which is the difference between the selected mean and the original mean divided by the relation to the square root of the phenotypic variance. So then you get this different form of the breeder's equation or the response to selection, which is related to additive variance and heritability. You can get from there to there by doing a little bit of math. And then you substitute, so you have this from the last slide, you substitute the variance that are explained, the variance that, or the markers the variance that explained by the marker you put that in instead of the additive variance, and you assume that the marker character has a heritability one. There's no uncertainty about the markers. So that's Mendelian inheritance. And so you get this equation. So that's a predicted response to marker-based selection. And then the relative efficiency of marker-based selection is, this is what we come down to, where you have, so it's the ratio between the variance explained by the marker and what's actually explained by the additive effects divided by the square root of heritability. So if you put that in a graph and where you have different, where you put different heritabilities in there, so you can see that as you go up here, so the marker explains more and more of the total variance, then the efficiency of incorporating of marker or using marker-assisted selection goes up and up, and also with lower heritability, it also increases quite significantly. So lower heritability, and the more that the marker explains, that's obvious, the more efficient marker-assisted selection is going to be. So as I said, marker-based selection is most effective when the heritability is low and when the variance explained by the markers are approaching the variance explained by additive variance. And so the power to detect QTLs actually... to detect QTLs decreases when heritability decreases. And so the variance explained by marker is likely to be high when heritability is high. So that's not really so useful. So a possible solution is to estimate the QTL when heritability is high, and then do subsequent marker-assisted selection when heritability is on populations where heritability is low. So there's a little bit of a conundrum there. So I'll now go on to some examples that I have taken from work that has been carried out at IBES or in collaboration with other institutes. So the first one goes back to some work we did on ryegrass. I'm sorry, it's not about red clover or alfalfa, but this is sort of the best example I could find. So some work was done in the 2000s that showed that identified some QTLs using populations that we had available at IBES, and also identified another one, or QTLs, both for heading date and for seed set. So we then got some money to carry out an experiment whereby we tried to. So we had a marker where we had A and B alleles, and the B allele was associated with sort of later or higher seed set, and also associated with flowering time. So the A and B alleles had opposite effects. So what we try to do, you don't need to sort of take much notice of the numbers in little size, but we created we created 9 different synthetic populations from ryegrass mother plants of the breeding population, where we sort of tried to make sure that the frequency of the A allele and the B allele changed gradually. So you went from here, where it was only the A allele present, and then a higher and higher percentage until you get up to this one here, where it's only B alleles, and then you had a sort of a kind of a control line where there was a little bit of a mixture of different alleles in there. So that scheme was divided by our senior plant breeder then, Alan Lovett. He's now retired, and he carried that out. And this is sort of the result of those crossings. So we created synthetic populations with different content of the key alleles for this flowering time gene. And so the percentage of the BB genotype goes up from 0 to 100, and the A allele frequency goes down from 100 to 0 in those populations. So it's quite a useful population to try and check to see whether there is some association with these traits that we were talking about, seed set in particular. That was what we really were interested in this particular project. So the expectation is, to recap, the BB genotype is associated with lower seed set and later heading date. And then if you go, this is just sort of screenshots from results of agronomic evaluations with various control varieties. And then you have the list of these synthetic populations, and there's no big, in terms of dry matter yield, they still perform well and comparable to commercial varieties. And also in terms of seed yield, there is a little bit of a, in 2 years here, there's an increase in seed yield in kilograms per hectare, a tendency to increase, certainly in 2015, as you increase the B allele frequency. So if you go to seed set, oh no. Yes. Yeah, so that's basically a graph showing the seed yield in relation to where the different varieties or the synthetic populations are. So there's a slight increase in seed yield in kilograms per hectare as you go along, but it's not a significant correlation. So it's a little bit all over the place. So if you then go to seed yield in seeds per square meter, then there is a correlation. It goes down from where the A allele is 100% to, or the B allele or B genotype is 0 and up to 100%. So if you then if you plot that in, there's certainly a relationship here between seeds per square meter and the BB genotypes. And if you plot that against the synthetic populations, you do get a little bit of a correlation that is significant. So as you increase the BB genotype, the seed yield in seeds per square meter goes down. So that was as far as we got with that. So it is kind of useful in that example. So the seeds in seed per square meter, but there was no real correlation with flowering time. I haven't really shown you that data. So it's difficult, but I just thought I would show it because I thought that was quite a good experiment to try and actually follow through whether the markers you initially identify in a QTL are actually useful. So it's a very mixed picture in this example here. If you go on to, yes, one last slide where I have shown the correlations between the different traits and the BB genotype. So that's just a recap. Seed number, there's a negative correlation with the BB genotype and positive correlation. So I'm not really sure what to make of this, but there's certainly a negative correlation between seed number per square meter and the BB genotype, and that was what we were predicting. Then I'll go on to another example, which is concerned with peas, and that's a nice project that's called PGene that David Lloyd is involved with people in collaboration with people from John Innes Center, particularly Claire Dominey. And now they are trying to create a new line where they have accumulated a total of 5 mutants that are mutated for visilin A. So these are seed proteins as far as I understand it. You can correct me if I'm wrong, David. Seed proteins, so visilin A, B, C, and D, and then convisilin something. So that sounds like a tall order, and those mutations were made using methodology that they have at the John Innes Center. And the next step is to get the mutations into a commercially useful line to try and see if those, I think they're undesirable seed proteins, and then hope that they get replaced by good seed protein, something like that. Anyway, this is more to do with the markers. So we have the mutant parent, you cross it with a commercial variety and you get your F1. And then you have, so the F1 is always hemizygous, so there's there's nothing there because of the mutation, and you self-pollinate the hemizygous F1 and produce your 1 to 2 to 1 ratio. And then you use marker-assisted selection. They have TACMAN markers associated with those mutations. And you screen the progeny, identify your lines, and you then keep your nulli-zygous progeny. And you cull the rest and take the lines forward to crossing with lines that are nulli-zygous for the other genes. And you try to pyramid those mutations into a single line. So that's the basic strategy. You have your commercial parent, which has this yellow star for the locus in question. Your mutant parent in red, and then yourself, you get the hemizygous, and you select only the ones that are in the F2 that have the mutation in there in a homozygous state, and you use your TACMAN to select that at an early stage. And then you pyramid those, you go on to the other mutations, and you cross different variants of those into F1s, and you self them, and you get accumulation of these nulli-zygous mutations, and you cull the rest. And for example, here they've selected one where they have accumulated 3 mutations and taken that forward. The rest have been ditched. And then this is sort of the current state of play with that project. So they have nulli-zygous for visilin B, visilin C, and visilin D, and then a nulli-zygous for some of it is overlapping, but there is a different one there in the 5th one for convisilin V. And so you have progeny. That's where they are at the moment. That looks like this, where you have 3 nulli-zygous mutations and 1 hemizygous. And so that'll be selfed to create that null version of visilin D. So that's quite an ambitious project, but one which that's well underway and likely to produce results. And in the end, they want to accumulate that into a commercial variety that hopefully have good protein, seed protein, and not the bad versions. So the other thing I'd like to mention briefly is particularly associated with outbreeding crops where you are working on synthetic populations and quite often working with half-zip progeny. Because you are losing quite a lot in half-zip progeny selection. You're using quite a lot of your genetic variation because you don't know the pollen donor, so you don't know. So you're working on sort of an average of all pollen donors when you take the progeny from your mother plant. So you lose a little bit of the genetic variation you could potentially work with. So that has been addressed by people in different places. There was it has been around to do what's it called? Paternity testing. That was the word I was looking for. But particularly for outbreeding crops, it's quite a useful thing if you can identify the pollen donors the pollen donors that contributed genetic material to your mother plants, and also the other work that has been done, particularly specifically on red clover, is here, and some of the people here are in the room, so you can ask them specific questions. But that's a useful thing to use where markers can actually do some good as well, particularly SSR markers, because you don't need very many to make sure that you identify your pollen donor. So I think that's a very nice example of where markers have been utilized to some good effect. And then I will now go on to GRAS briefly, and further on genomic selection. Now, if you in the past or up to now, linkage mapping has been used a lot to identify QTLs, but there are some limitations. It requires growth of 3 generations and quite large populations for sufficient resolution to identify a narrow region where your QTL is, and also the big thing is that when you identify QTLs in a specific populations, it may not be particularly relevant for your breeding population. It may be specific for that cross, and when you try to identify it in the breeding population, it may not be there. So that's a well-known argument for the limitation of using mapping populations. And then association or linkage disequilibrium mapping is something that came on the scene in the beginning of this century, I think, particularly in plants. So it is to identify patterns of polymorphism that vary systematically between genotypes with contrasting phenotypes in more or less random populations, potentially. So on the left you have linkage mapping, and so that's the traditional version of creating F2 and then recombinant inbred lines, and then you get sort of a QTL resolution. That's sort of in that region, perhaps as an example. But then association mapping is sort of relying on some effect of a founder population that was theoretically generated many generations ago, and then you have this SNP polymorphism that explains some trait, and then as the recombinations through the generations carry on, the region around that QTL or where that trait is becomes narrower and narrower. So as you many generations ahead, you have the potential to take your population and get a very narrow region, so a very high resolution of your QTL compared to linkage mapping. So that's the sort of the theory of it, the basic theory of it. But I will then sort of go on to explain a little bit about linkage disequilibrium. You have to forgive me if it's a bit too basic for you, but there we are. So you have complete, what is linkage disequilibrium? So it's a non-random association between alleles at 2 loci. So it's a difference between the probability of those 2 loci occurring together versus the independent probability of them. So this is complete disequilibrium, where you have markers and different chromosomes that are completely associated. But here on the right, you have complete equilibrium, where you have sort of a random or a random association between the alleles at the different loci. So the measure of LD, the basic measure was on the previous slide, but that's dependent on allele frequencies, so that can vary quite a lot. So you ideally want something that sort of varies between 0 and 1. So people have devised various different versions of measures of linkage disequilibrium. There's this D prime that's described here. And then there's the one that I guess people use mostly, which is the correlation between the squared D and the product of all the 4 potential alleles at the 2 loci. And theoretically, LD in sort of an idealized population, LD decays. Decay is related to the number of generations and the recombination frequency. So the lower the recombination frequency is, i.e. the closer the marker is to the trait, or the 2 markers are to each other, the longer it takes for linkage disequilibrium to decay. And of course, if they're incomplete, if they're not linked at all, it's very fast. It goes down to 0. So a little bit more linkage disequilibrium. So that's another way of describing the complete linkage disequilibrium. So you have this little table here with, and this is the other situation where you have complete linkage equilibrium, where R squared is 0, and this D prime, the absolute value of that, is 0. So if you then go on to, so this is sort of the theoretical situation that never happens in reality, where you have association with this very naive illustration of what you sort of are looking for. So you have a marker that's associated with lower biomass or something, and whatever trait you're interested in, and another one associated with high biomass. So that's what you want to, that's what you're aspiring to get or trying to get. And if you then go to an example I've shown here, where you have linkage disequilibrium, where the R squared value is 0.25, which is not particularly high, but not 0 either. So if you, for example, have a marker, that's this one here, or if this is a QTL, say, that explains 10% of total phenotypic variance, so it's not the causative SNP, and marker 2 explains 25%, so because R squared is 0.25. So it then only explains 2.5% of the total phenotypic variance. So that's another sobering lesson, that if you have a marker in not too closely in linkage disequilibrium with your QTL, then the association dips quite dramatically. So that requires a large sample size for sufficient power, or a higher value of R squared ideally. So that's another way of, that's another little illustration of what you ideally require in terms of marker density if you want to do genome-wide association studies. Say, for example, we want an R squared value of 0.5 to allow us for reasonable power. And say you were working with red clover, the genome size is around there, 420 mega, bases, and say LD decays to 0.5 and 10 kb. I think that might be optimistic, but let's just say that for argument's sake. We would ideally need a marker for every 20 kb because there are 10 kb to either side, and so you would sort of ideally need 21,000 evenly spaced markers. So that's a sort of a very theoretically based ballpark figure of the number of markers you might need. And the larger the genome is, and the more markers you need, of course, but also the decay, the rate of decay in different species varies. So it's obviously decays very rapidly in outbreeding species and much slower in inbreeding species. But then the resolution is lower when the decay takes longer. So that's another thing to think about when you do GRAS. The other thing is structure in the populations. So if you have cases, for example, which I've taken this from sort of human genetics, where they usually work with cases and controls. So somebody, a population where you want to look at the risk of a disease, a genetically based disease, then you worry about whether cases are disproportionately represented in one population, in one subgroup than in another. And then you get skewed, you get sort of a bias and false positives. So that's also something you have to take great care of when you evaluate genome-wide association studies. But most software programs can easily take that into account in some form, and I'm sure people will come back to that later in the course. So this is just an illustration of what of the importance of it. So if you want to evaluate genotype-phenotype associations, you have to assess the population structure and incorporate that into your analysis. And there are various ways of doing it. Using the principal component analysis of markers that are distributed all over the genome, so that there are programs called EigenStrat or EigenSoft, I think. So people either use that or structured association, which is sort of a Bayesian-based, and I'm sure some of you are familiar with that. And you can use the sort of the fraction of they sort genotypes into whether they belong into different population groups, and so you get a sort of a fraction from 1 to 0. But then you have to make sure that you have you are choosing the right number of subgroups. So it's not always straightforward to use that. So you usually eliminate rare polymorphisms, and then you test your association using a chi-squared analysis. If your trait is binary, that's quite rare in plant breeding. And then more normally you would use ANOVA or regression if your trait is quantitative. And then you must also take account of and correct for multiple tests, because if you use a nominal probability cutoff of 0.05 for every 100, you detect that are potentially significant. So 20 of those will be potentially false positives. So there are also ways of correcting for and to cut down on the number of false positives. And this is a little example I'll go through quickly. Am I going over time, or? Okay, right. Then you go to flowering time in red clover, where we had this panel, European and a few populations from Asia, and 600 plants from 70 populations. must remember this. So, and then we used the TASL program, which I'm sure some of you have heard of, and we have 8,000 SNPs. So that's potentially not enough, but we did that anyway, and then we found one marker that was associated with flowering date. We were looking for other traits as well, but didn't find any real associations. But one gene there, and that's located on chromosome 3, and seems promising in terms of the identity of the gene. So it's a homologue of the VEG2 gene, a FT homologue from P, and it has an effect in our populations. So genomic selection, quickly. That's where I guess markers are coming into their own, because you don't need, you use the effect of all the markers whether or not they're significant, and that's the difference between that and GWAS. And you use the combined effect of all the markers in an individual to calculate the breeding value, and then you the breeding value is related to what combination of markers an individual will give to its progeny. And you need a training population to make that prediction model, which is sort of symbolized here. You will hear much more about that later on. And the training population has phenotypic as well as genotypic data. And so you put those two data together, and you calculate a marker effect of the training population, and you use that to predict the genomically estimated breeding value potentially in your validation population. So that's the basic principle. As I to recap, put the genotype and phenotype data together, and you make predictions of breeding values in your validation population, and then you select your candidates on the basis of those genomically estimated breeding values. So, to recap, the combined marker effect are used to estimate the breeding values of an individual, and the accuracy of the predictions is tested on the training population using cross-validations, and if they're reasonable, you get your genomically estimated breeding value, and you use that. And so it's useful for traits that are expensive or difficult to measure, or those that are measured late in the life cycle. You only need the DNA, and it can potentially save money or time or both. These are some results from our grass breeding program, where we have now a training population size of 825. I think there has been a lot of talk about different types of models, and we used a program which evaluated the accuracies on different types of models. You probably heard of them, many of you, but none of them really beats the basic RR-BLUP method that I think also will be the mainstay of the rest of this course, but I don't know. So the RR-BLUP is I guess, is the one that people use most. So these are different traits: protein, water-soluble carbohydrates, dry matter digestibility, and non-detergent digestibility, and forage yield in year 1. So the accuracy of the yield is still lower than in the quality traits, and that's something that's been quite consistent in the grass breeding program that we are working on at IBIS. And so this is an example of where we have used a program that predicts the variance and the mean value of, or the variance and the mean superior progeny value was the word I was looking for. And we have used mother plants, 4 different mother plants in the breeding population, going back to see how well would it actually have performed, how well did it predict different traits in the progeny. And if you go to water-soluble carbohydrates, which is a character that has been very dear to us in the breeding program, and one we have put a lot of weight on, or the breeders have put a lot of weight on. So when you can see the progeny is predicted to sort of be at the high end, there's a little bit of an exception there, so we're a little bit middling there. And in contrast to that, nitrogen or protein is kind of inversely correlated with water-soluble carbohydrates. So the prediction of the crosses that these mother plant can make potentially in that generation is a bit on the lower side. So that sort of has gave us confidence that it is actually working quite well in predicting the progeny performance and the variance of them. So all the potential outcomes are this sort of gray cloud, and so the purple ones are the ones that are only possible with that mother plant. So that's how it works, and this is taken from this program called POPVAR that we have been using lately in our work. And I think that's it. Yeah, thank you very much for your attention. Sorry it dragged on a bit.
Thank you, Leif.
Questions?
Yeah. There is time for questions. Are there any questions from the audience, please? Quite interesting presentations.
I am here all through the course, so yeah, come up and ask me if there is.
Yes.
Thank you for the presentation. I hear that in case of complex traits, like for example abiotic stress, there are QTLs associated. I can found in the literature, but I can also see that the effect is very minor for those QTLs. Do you have any suggestions that are those QTLs valuable to use? And if not, how can we improve the value of those QTLs, which are associated actually, but with little effect?
Well, in genomic selection, for example, it doesn't. The marker doesn't have to be, each individual marker doesn't have to be significantly associated, but so it just uses all the QTLs that are there potentially. I'm not sure that was what you were asking, but
you say that all the markers are valuable if even they don't have a high effect. I mean,
well, theoretically, if you have, for example, a QTL that, or a trait that's explained by 50 QTLs, and they have different effect. But if you have a marker that explains a high percentage of each of those QTLs, then they might be useful in combination. So does that help?
Thank you. And also you show the number of studies that are going to increase with the passage of time for QTL detections. But I don't know if all those QTLs are already applicables. There are lot and lot studies for the discovery of QTLs, but for the applications, there are not many studies in that direction. So what you suggest, or what is your opinion for that?
I didn't really get the question.
Because the number of studies you have shown in the slide, first slide, that there are too many studies which are directed towards the discovery of QTLs. But I don't see that there are too many studies who also apply the QTLs in breedings.
No, that's exactly my point. Yeah, yes.
So
yes.
what is the negative side? Why they don't apply the QTLs? Because they have been discovered already. They should be applied in the practical side.
Yes, I guess we're all guilty of that. QTLs are discovered, and then you have to do more work to validate them in different populations, and that's quite often a lot of effort. I mean, for example, that work I showed with ryegrass, and where we had to make all these combinations, that was a big undertaking, and it didn't really, the effect was not fantastic in the end. I guess it's because it requires a lot of effort to follow that up and really see if the QTL is useful in your breeding population. But I guess if you have a breeding population and you have a QTL, the first thing we would do is to look at whether the allele is fixed in your breeding population for one trait or the other, for one extreme or the other. And then if it's fixed for the good allele, then you don't need to do anything more. But if it's fixed for the bad one, you might consider wanting to gradually increase the frequency of the good allele, so to speak. But yeah, I think we're all guilty of it. There are QTLs collecting dust in the literature. And
for the young researchers, do you suggest that we should go towards the validation studies instead of discovery of new QTLs?
Well, I guess if you can work as much as possible on breeder-relevant material, I guess that's the thing that has been most useful in at our institute. We have the freedom to work directly with the breeding populations that are there. So. But I also appreciate if you want to get segregation of a trait, you have to go to maybe some exotic material and find extremes, and you want to cross them and look at the underlying mechanism. So it's not just about breeding. I guess you also want to find some underlying mechanisms. So.
Yeah, just maybe to add something on this topic. I think there are many examples where QTL are used now, but they are not published. It's in breeding company, and they are using for QTL with quite large effects, but it's used now, but it's not published. It's the difference. And what is published, I agree with Leif, that it will be for more biological understanding on the effect of the gene physiologically. And it's a different topic. But I think QTLs are used already in breeding program. When you look in maize or in wheat or in different programs. QTLs plus genomic selection.
Yes, I agree with that, yeah.
Another question? Yeah, Bernadette.
Yes, Leif, could you come back to your last slide, because I didn't catch the x-axis and the y-axis. Yes, this one. Could you explain more this slide?
Okay. We looked at this program called PopVar, which has been used which predicts the performance of progeny from all potential pairwise combinations that you would want to look at. So although it was designed and used in inbreeding crops, I think mainly for barley or wheat, we tried to see how it performed in ryegrass by going back to a previous generation of our breeding program. And there we had these 4 mother plants. And those 4 mother plants were used to generate the space plant nursery. The half the progeny from those were used in the space plant nursery for the following generation. So we wanted to, it's sort of a retrospective fit to see how well the genomic selection would have performed if we had used it then. And so there are a number of potential combinations that those 4 mother plants could have made when they went into the pollen chambers. And then we have highlighted those in purple, and then in gray there's the cloud of all potential combinations, which is over, in that particular case, it's 4,100 and something. And for these, there are 53 potential combinations with those here. And so we went into to look at water-soluble carbohydrates, which I said is a trait that we emphasized quite a lot in the selection, and then try to contrast that with nitrogen or protein. And so we looked at, we have the superior progeny value, which is sort of the 10% of the best potential means, and then you have the variance that has to do with the difference in allele frequency between the different combinations of parents. And so the upshot of that is that and I can document that with numbers, but this was just to show the graph, that the mean value of all the potential combinations of these 4 mother plants in terms of water-soluble carbohydrates is sort of on the high side of the spectrum, and it's a little bit on the low side, particularly in these 2 mother plants when it concerns nitrogen, which we measure with near-infrared spectroscopy. So it was just an illustration of what it would have looked like if we had used it then. So and this was indeed what happened then. There was great emphasis on getting improvement in water-soluble carbohydrates and generate more of these high sugar grasses. So is that?
More questions? So if not, thank you everybody. Thank you, Leif.